Venus flytrap extract is available on the market as an herbal remedy, sometimes as the prime ingredient of a patent medicine named carnivora. According to the American Cancer Society, these products are promoted in alternative medicine as a treatment for a variety of human ailments including HIV, Crohn's disease and skin cancer, but available scientific evidence does not support the health claims made for Venus flytrap extract. The Venus flytrap is a carnivorous plant native to subtropical wetlands on the east coast of the United States. In North Carolina and South Carolina it catches its prey, chiefly insects and arachnids, with a trapping structure formed by the terminal portion of each of the plant's leaves, which is triggered by tiny hairs called trigger hairs, or sensitive hairs, on their inner surfaces. When an insect or spider crawling along the leaves contacts a hair, the trap prepares to close, snapping shut only if another contact occurs within approximately 20 seconds of the first strike. Triggers may occur with a tenth of a second of contact. The requirement of redundant triggering in this mechanism serves as a safeguard against wasting energy by trapping objects with no nutritional value, and the plant will only begin digestion after 5 more stimuli to ensure it has caught a live bug worthy of consumption. Dionea is a monotypic genus closely related to the waterwheel plant Aldrovanda vesiculosa, and Sundus drosera, all of which belong to the family Droseraceae. Although widely cultivated for sale, the population of the Venus flytrap has been rapidly declining in its native range. The species is currently under Endangered Species Act review by the U.S. Fish and Wildlife Service. Historically, the plant was also known by the slang term tippity-witchet, or tippity-twitchet, possibly an oblique reference to the plant's resemblance to human female genitalia. The term is similar to the term tippet de witchet, which derives from tippet and witchet, archaic term for vagina. In contrast, the English botanist John Ellis, who gave the plant its scientific name in 1768, wrote that the plant name tippity-witchet was an indigenous word from either Cherokee or Catawba. The plant name, according to the Handbook of American Indians, derives from the Lenape word tidipawitchik, they leaves which wind around or involve. The Venus flytrap is a small plant whose structure can be described as a rosette of 4 to 7 leaves, which arise from a short subterranean stem that is actually a bulb-like object. Each stem reaches a maximum size of about 3 to 10 centimeters, depending on the time of year. Longer leaves with robust traps are usually formed after flowering. Flytraps that have more than 7 leaves are colonies formed by rosettes that have divided beneath the ground. The leaf blade is divided into 2 regions: a flat, heart-shaped photosynthesis-capable petiole, and a pair of terminal lobes hinged at the midrib, forming the trap, which is the true leaf. The upper surface of these lobes contains red anthocyanin pigments and its edges secrete mucilage. The lobes exhibit rapid plant movements, snapping shut when stimulated by prey. The trapping mechanism is tripped when prey contacts one of the 3 hair-like trichomes that are found on the upper surface of each of the lobes. The mechanism is so highly specialized that it can distinguish between living prey and non-prey stimuli, such as falling raindrops. 2 trigger hairs must be touched in succession within 20 seconds of each other, or 1 hair touched twice in rapid succession, whereupon the lobes of the trap will snap shut, typically in about 1/10 of a second. The edges of the lobes are fringed by stiff hair-like protrusions or cilia, which mesh together and prevent large prey from escaping. These protrusions, and the trigger hairs, also known as sensitive hairs, are likely homologous with the tentacles found in this plant's close relatives, the sundews. Scientists have concluded that the snap trap evolved from a flypaper trap similar to that of Drosera. The holes in the meshwork allow small prey to escape, presumably because the benefit that would be obtained from them would be less than the cost of digesting them. If the prey is too small and escapes, the trap will usually reopen within 12 hours. If the prey moves around in the trap, it tightens and digestion begins more quickly. Speed of closing can vary depending on the amount of humidity, light, size of prey, and general growing conditions. The speed with which traps close can be used as an indicator of a plant's general health. Venus flytraps are not as humidity dependent as are some other carnivorous plants, such as Nepenthes, Cephalotus, most Heliamphora, and some Drosera. The Venus flytrap exhibits variations in petiole shape and length and whether the leaf lies flat on the ground or extends up at an angle of about 40 to 60 degrees. The 4 major forms are typica, the most common, with broad decumbent petioles; erecta, with leaves at a 45-degree angle; linearis, with narrow petioles and leaves at 45 degrees, and Filiformis, with extremely narrow or linear petioles. Except for filiformis. All of these can be stages in leaf production of any plant depending on season, decumbent in summer versus short versus semi-erect in spring, length of photoperiod, long petioles in spring versus short in summer, and intensity of light, wide petioles in low light intensity versus narrow in brighter light. The plant also has a flower on top of a long stem, about 6 inches long. The flower is pollinated from various flying insects such as sweat bees, longhorn beetles and checkered beetles. The Venus flytrap is found in nitrogen and phosphorus-poor environments, such as bogs, wet savannas, and cannebraches. Small in stature and slow-growing, the Venus flytrap tolerates fire well and depends on periodic burning to suppress its competition. Fire suppression threatens its future in the wild. It survives in wet sandy and peaty soils. Although it has been successfully transplanted and grown in many locales around the world, it is native only to the coastal bogs of North and South Carolina in the United States, specifically within a 100-kilometer radius of Wilmington, North Carolina. One such place is North Carolina's Green Swamp. There also appears to be a naturalized population of Venus flytraps in northern Florida as well as an introduced population in western Washington. The nutritional poverty of the soil is the reason it relies on such elaborate traps. Insect prey provide the nitrogen for protein formation that the soil cannot. They tolerate mild winters, and Venus flytraps that do not go through a period of winter dormancy will weaken and die after a period of time. They are full sun plants, usually found only in areas with less than 10% canopy cover. The habitats where it thrives are typically either too nutrient poor for many noncarnivorous plants to survive, or frequently disturbed by fires which regularly clear vegetation and prevent a shady overstory from developing. It can be found living alongside herbaceous plants, grasses, sphagnum, and fire-dependent arundinaria bamboos. Regular fire disturbance is an important part of its habitat, required every 3 to 5 years in most places for D. machipula to thrive. After fire, D. machipula seeds germinate well in ash and sandy soil, with seedlings growing well in the open post-fire conditions. The seeds germinate immediately without a dormant period. Dionea machipula occurs naturally only along the coastal plain of North and South Carolina in the U.S., with all known current sites within 90 kilometers of Wilmington, North Carolina. A 1958 survey of herbaria specimens and old documents found 259 sites where the historical record documented the presence of D. machipula, within 21 counties in North and South Carolina. As of 2019, it was considered extirpated in North Carolina in the inland counties of Moore, Robeson, and Lenoir, as well as the South Carolina coastal counties of Charleston and Georgetown. Remaining extant populations exist in North Carolina in Beaufort, Craven, Pamlico, Carteret, Jones, Onslow, Duplin, Pender, New Hanover, Brunswick, Columbus, Bladen, Sampson, Cumberland, and Hoke counties, and in South Carolina in Horry County. A large-scale survey in 2019, conducted by the North Carolina Natural Heritage Program, counted a total of 163,951 individual Venus flytraps in North Carolina and 4,876 in South Carolina, estimating a total of 302,000 individuals remaining in the wild in its native range. This represents a reduction of more than 93% from a 1979 estimate of approximately 4,500,000 individuals. A 1958 study found 259 confirmed extant or historic sites. As of 2016, there were 71 known sites where the plant could be found in the wild. Of these 71 sites, only 20 were classified as having excellent or good long-term viability. Most carnivorous plants selectively feed on specific prey. This selection is due to the available prey and the type of trap used by the organism. With the Venus flytrap, prey is limited to beetles, spiders and other crawling arthropods. The Dionea diet is 33% ants, 30% spiders, 10% beetles, and 10% grasshoppers, with fewer than 5% flying insects. Given that Dionea evolved from an ancestral form of Drosera, carnivorous plants that use a sticky trap instead of a snap trap, the reason for this evolutionary branching becomes clear. Drosera consume smaller, aerial insects, whereas Dionea consume larger terrestrial bugs. Dionea are able to extract more nutrients from these larger bugs. This gives Dionea an evolutionary advantage over their ancestral sticky trap form. The Venus flytrap is one of a very small group of plants capable of rapid movement, such as Mimosa pudica, the telegraph plant, starfruit, sundews, and bladderworts. The mechanism by which the trap snaps shut involves a complex interaction between elasticity, turgor, and growth. The trap only shuts when there have been two stimulations of the trigger hairs. This is to avoid inadvertent triggering of the mechanism by dust and other wind-borne debris. In the open, untripped state, the lobes are convex, bent outwards, but in the closed state, the lobes are concave, forming a cavity. It is the rapid flipping of this bistable state that closes the trap, but the mechanism by which this occurs is still poorly understood. When the trigger hairs are stimulated, an action potential, mostly involving calcium ions, see calcium in biology, is generated, which propagates across the lobes and stimulates cells in the lobes and in the midrib between them. It is hypothesized that there is a threshold of ion buildup for the Venus flytrap to react to stimulation. The acid growth theory states that individual cells in the outer layers of the lobes and midrib rapidly move 1H+ hydrogen ions into their cell walls, lowering the pH and loosening the extracellular components, which allows them to swell rapidly by osmosis, thus elongating and changing the shape of the trap lobe. Alternatively, cells in the inner layers of the lobes and midrib may rapidly secrete other ions, allowing water to follow by osmosis, and the cells to collapse. Both of these mechanisms may play a role and have some experimental evidence to support them. Flytraps show an example of memory in plants. The plant knows if one of its trigger hairs have been touched, and remembers this for a few seconds. If a second touch occurs during that time frame, the flytrap closes. After closing, the flytrap counts additional stimulations of the trigger hairs, to 5 total, to start the production of digesting enzymes. If the prey is unable to escape, it will continue to stimulate the inner surface of the lobes, and this causes a further growth response that forces the edges of the lobes together, eventually sealing the trap hermetically and forming a stomach, in which digestion occurs. Release of the digestive enzymes is controlled by the hormone jasmonic acid, the same hormone that triggers the release of toxins as an anti-herbivore defense mechanism in non-carnivorous plants. Once the digestive glands in the leaf lobes have been activated, digestion is catalyzed by hydrolase enzymes secreted by the glands. One of these enzymes includes GH18 chitinase, which breaks down chitin-containing exoskeleton of trapped insects. Synthesis of this enzyme begins with at least 5 action potentials, which will stimulate transcription of chitinase. Oxidative protein modification is likely to be a pre-digestive mechanism used by Dionaea mashipula. Aqueous leaf extracts have been found to contain quinones such as the naphthaquinone plumbagon that couples to different NADH-dependent diaphoruses to produce superoxide and hydrogen peroxide upon autooxidation. Such oxidative modification could rupture animal cell membranes. Plumbagon is known to induce apoptosis, associated with the regulation of the BCL2 family of proteins. When the Dionaea extracts were pre-incubated with diaphoruses and NADH in the presence of serum albumin, subsequent tryptic digestion of SAW was facilitated. Since the secretory glands of Droseraceae contain proteases and possibly other degradative enzymes, it may be that the presence of oxygen-activating redox cofactors function as extracellular pre-digestive oxidants to render membrane-bound proteins of the prey, insects, more susceptible to proteolytic attacks. Digestion takes about 10 days, after which the prey is reduced to a husk of chitin. The trap then reopens, and is ready for reuse. Plants can be propagated by seed, taking around 4 to 5 years to reach maturity. More commonly, they are propagated by clonal division in spring or summer. Venus flytraps can also be propagated in vitro using plant tissue culture. Most Venus flytraps found for sale in nurseries' garden centers have been produced using this method, as this is the most cost-effective way to propagate them on a large scale. Regardless of the propagation method used, the plants will live for 20 to 30 years if cultivated in the right conditions. Venus flytraps are by far the most commonly recognized and cultivated carnivorous plant, and they are frequently sold as houseplants. Various cultivars, cultivated varieties, have come into the market through tissue culture of selected genetic mutations, and these plants are raised in large quantities for commercial markets. The cultivars, Akai Ryu and Southwest Giant, have gained the Royal Horticultural Society's Award of Garden Merit. The Venus flytrap is only found in the wild in a very particular set of conditions, requiring flat land with moist, acidic, nutrient-poor soils that receive full sun and burn frequently in forest fires, and is therefore sensitive to many types of disturbance. A 2011 review identified 5 categories of threats for the species: agriculture, road building, biological resource use, poaching and lumber activities, natural systems modifications, drainage and fire suppression, and pollution. Fertilizer. Habitat loss is a major threat to the species. The human population of the coastal Carolinas is rapidly expanding. For example, Brunswick County, North Carolina, which has the largest number of Venus flytrap populations, has seen a 27% increase in its human population from 2010 to 2018. As the population grows, residential and commercial development and road building directly eliminate flytrap habitat, while site preparation that entails ditching and draining can dry out soil in surrounding areas, destroying the viability of the species. Additionally, increased recreational use of natural areas in populated areas directly destroys the plants by crushing or uprooting them. Fire suppression is another threat to the Venus flytrap. In the absence of regular fires, shrubs and trees encroach, outcompeting the species and leading to local extirpations. D. Mashipula requires fire every 3 to 5 years, and best thrives with annual brush fires. Although flytraps and their seeds are typically killed alongside their competition in fires, seeds from flytraps adjacent to the burnt zone propagate quickly in the ash and full sun conditions that occur post a fire disturbance. Because the mature plants and new seedlings are typically destroyed in the regular fires that are necessary to maintain their habitat, D. Musipula's survival relies upon adequate seed production and dispersal from outside the burnt patches back into the burnt habitat, requiring a critical mass of populations, and exposing the success of any one population to metapopulation dynamics. These dynamics make small, isolated populations particularly vulnerable to extirpation, for if there are no mature plants adjacent to the fire zone, there is no source of seeds post-fire. Poaching has been another cause of population decline. Harvesting Venus flytraps on public land became illegal in North Carolina in 1958, and since then a legal cultivation industry has formed, growing tens of thousands of flytraps in commercial greenhouses for sale as household plants. Yet in 2016, the New York Times reported that demand for wild plants still exists, which has led to a Venus flytrap crime ring. In 2014, the state of North Carolina made Venus flytrap poaching a felony. Since then, several poachers have been charged, with one man receiving 17 months in prison for poaching 970 Venus flytraps, and another man charged with 73 felony counts in 2019. Poachers may do greater harm to the wild populations than a simple count of individuals taken would indicate, as they may selectively harvest the largest plants at a site, which have more flowers and fruit and therefore generate more seeds than smaller plants. Additionally, the species is particularly vulnerable to catastrophic climate events. Most Venus flytrap sites are only 2 to 4 meters, 6.5 to 13 feet, above sea level and are located in a region prone to hurricanes, making storm surges and rising sea levels a long-term threat.